Good morning, students. I am Dr.S.Vinod Kanna, Assistant Professor at Botany, Department of Botany, School of Science, Tamil Nadu Open University. Today, I am going to take the undergraduate program second year course of microbiology and plant pathology. The course comprises five blocks, four blocks with microbiology and final block with plant pathology. Today, I am going to take the first segment of 2 units of microbiology. The course code is BBOTS 32. The microbiology and plant pathology is paper. This course is coming under third semester, and this now I am going to take the history of microbiology and the introduction for microbiology. Students, you may all know that microbiology is the study of microbes, the microorganisms and its life are elaborately discussed in this paper. So, what is microbiology? I already told you that what is microbiology. The microbes, the organisms, the living organisms is able to see under microscope only. They are unable to see the naked eyes. So these such type of organisms are exist in the world many more years from the major portion of this microbiology is comprised usually microscopic organisms. Some organisms are large through some are animals like helminths, worms. The subgroups of microbes they are bacteria, archaea, fungi, protozoans, algae, viruses, multicellular animals or parasites like helminths. These are all the sections that comes under microbiology. We can see the slide of my bacteria is the photograph is taken from the electron microscope. You can see rod like structure of bacteria. This is the rod like bacteria. Commonly we can call it bacillus, like bacillus thuringiensis, bacillus subtilis. These kind of organisms comes under rod like bacteria. So usually bacteria are normally rod shapes, round shapes, coma shaped, and spiral shape. These are four shapes only in bacteria. The next organism group is fungi. Fungi you can see a mycelium. Mycelium is the portion and a body portion. You can see the electron microscopic view of a fungi. This is a penicillium. Penicillium. The next The next group is protozoans. Here you can see the microscopic view of amoeba and giardia. The next slide you can see the viruses. The left side you are seeing in the bacteriophage, right side is two viruses. The viruses are many shapes and its classification are many. This is a group of microbes. You can see electron microscope of it here. This is a very minute organisms in the world. Is viruses. The bacteriophage is phage viruses which only attacks on bacteria. You can see the helminths, the last group of microbes, helminths, which produce a lot of problems with the human beings. You can see the slide as caries and tapeworm. So disciplines of microbiology is you can the disciplines comes under microbiology is bacteriology which is the study of bacteria, mycology, study of fungi, parasitology, study of parasites, immunology in blood, immune system, epidemiology, biotechnology, virology, environmental microbiology, bioremediation. These are all the disciplines which comes under microbiology. Then we can go to the history of microbiology. When you take the history of microbiology, you can go beyond the discoveries of microbiology in the 17th century, hardly in 1600, in 1665, in 1665, English scientist whose name is Robert Hooke used to see a microorganism under a lens, which is a a primitive microscope at that stage. From that onwards, we developed a microscope. So with the three lenses and glasses, he used to see the microbes. And there are exactly two magnifying lenses in this microscope. Microscope reported that the life smallest units were little boxes. He reported the little boxes after seeing and the microbes under the microscope. So it's called cells. He named it cells. His work started the process of the development of a cell theory of life. After this, he proposed the cell theory. So, whatever we are seeing under microscope is the father is the pioneer was Robert who started to see the microbes under microscope. You can see the slide that is Hooke's microscope. You can see the two lenses here and sample here. You can see this viewpoint. This is the development of Robert Hooke's microscope. Next scientist we can see is Anton Leeuwenhoek. Exactly after 8 to 10 years, the first person who is observed living cells with a simple microscope. He is an amateur scientist and he he developed more than two lenses to see the living organisms under the microscope. He is the first to see the rod-shaped bacteria under the microscope, and he named it animalcules. In the slide, you can see Anthony van Leeuwenhoek. The next scientist you can see is Francisco Redi. In 1668, he opposed the prevailing theory of spontaneous generation, maggots in meats. He used to covered jars to show that maggots come from the flies. Earlier, earlier we can see Francisco Redi is opposed a theory of spontaneous generation. Earlier, what is called as a theory of spontaneous generation is existed. The spontaneous generation is means that the life originates from non-living to living. Automatically, it is the living things has been created. This is the theory existed earlier. He only Francisco Redi only opposed that prevailing theory of spontaneous generation by using a method called maggots meat. He used covered jars to show that maggots came from flies, strong evidence against spontaneous generations. He termed his theory is biogenesis. Life comes from life. This is the one-liner of biogenesis. Earlier, life comes from non-living. He opposed and broke that theory, and he proposed life comes from life. But issue of spontaneous generation was actively believed for many more years after. So this is the Francisco theory. He used to experiment with two beakers of meat, one tightly closed and the another one is open. After a period of time, the closed one having a life in on the surface of closer, but in the open one having the microbes on the meat on the meat itself. This is the theory, and he proposed this theory and he interpreted that life comes from a life. Next scientist is more important is Edward Jenner. Edward Jenner, who created the vaccines. Now, normally we can see very aware of normal people apart from microbiology, other subjects people also came to know that vaccine is important in nowadays in the pandemic situation. But this is discovered earlier by Edward Jenner in the 1970s. So, means 1700s in 18th century, starting of 18th century. He started to observe is made not affected by a disease which is exist on that particular time, and he isolated the sputum, that means the saliva of a maid, and started seeing this designing this vaccines for cowpox. Cowpox. And the milkmaids had cowpox lesions but not smallpox. He purposefully took scalpings from cowpox blisters and scrapped an 8-year-old volunteer with immaterial child with mild illness and not smallpox. This is the experiment he carried out. The lesions, from the lesions, he got that microbes and inserted and stuck to the 8-year-old volunteer and not getting affected by smallpox. From this experiment, he interpreted that that is to expel the virulence from a virus and give it to the patients to give resistance to the particular vaccine, the particular viral diseases. The vaccination comes from the Latin word vacca, meaning cow. Jenner laid the foundation for pasteurization. Later, work with other vaccines. This is the famous photograph of Edward Jenner giving at the volunteer of the lease himself, a cowpox. Next scientist is very famous, Louis Pasteur. Louis Pasteur is a Frenchman. He proposed the pasteurization of milk. The French scientists built the depth of flow to the spontaneous generation theory. He devised an ingenious curved-necked flask that prevented contamination air from reaching boiled beef broth. The broth remained uncontaminated even though exposed to the air. He used very lucky, no endosperms present in it, or it would have failed. He developed a process we call pasteurization. He heated wine to kill contaminating microbes, cured sick wine by heat treatment to kill pathogens in milk also. He proved that fermentation was possible by the microbe yeast. He developed vaccines for rabies, anthrax vaccines led to immunity disease and routinely killed many people. Used to help people long before they understood how they even worked. He began the revolution in science that led to the golden age of microbiology. He is the one who started this revolution of the microbiology. He is passionate. The photograph is shows in the PPT is Louis Pasteur. He is the one the basic laid the basic foundation for microbiology and bacteriology. You can see this is the bottleneck experiment. You can see uncontaminated wine. The next one is also important, very famous Robert Koch. He is parallel to Louis Pasteur. Robert Koch, who developed Koch Pasteur. Koch Pasteur is the concept of microbiology. It is a very, very important theory of microbiology, very prominent theory. He postulated, he derived these things from a series of experiments, important techniques. He is an important technician for determining the actual microbial cause agent of a disease. If a disease caused to a human or a plant, it is caused by a microbe, the organism which we named is causal organisms, which is responsible to create the disease for this particular animal or plant. He discovered the tuberculosis bug, Mycobacterium tuberculosis. He discovered the cause of anthrax, Bacillus anthracis, from blood and dead cattle. He cultured that bacteria in pure culture, injected bacteria in live cattle. He isolated a culture of bacteria from dead cattle to a artificial medium he developed, and he cultured that particular organism, that is Bacillus anthrax, and he injected that again into a living organism, cattle. This led to the establishment of procedure for determining microbial causes of disease. Then he, after injecting that microbes into a living cattle, that the cattle produced the lesion that that is same to the cattle who died earlier by this same microbe. From that lesion, he again isolated and pure cultured that the same bacillus. These 4 points are the concept of co-spatulating. The course and Pasteur's work helped establish the germ theory of diseases, that microorganisms cause death, diseases in the people, animals, and even plants. Then, then Ivanovsky in 1892 discovered that plant diseases can be caused by small organisms that were so small they passed through filters. Vapor mosaic virus was later identified as the cause, beginning of virology. These are earlier we saw this maximum of bacteria. Now Ivanovsky came and he started doing research on virus of plants. mainly the tobacco mosaic viruses. The beginning of virology. Today we have discovered new and weird things like virus strains. You can see this is the bacteriophage viruses under electron microscope. We can see the scale of 100 micrometer. You can see roughly 200 to 300 micrometer of bacteriophage is about length. You can see this picture under you can see this picture which can be able to see under electron microscope only. The dark side is Paul Eldridge, German doctor, want to find a magic bullet, an agent that would kill the disease agent without hurting the patients. Developed salvarsan from syphilis agent. This was the arsenical compound that was effective against syphilis. The syphilis is a kind of disease in human. Antimicrobial agent medicine to treat a microbial disease. It was chemical chemotherapy. You can see the organism under microscope is syphilis. In the right side, you can follow it. Next comes is Joseph Lister. In 1860s, British surgeon, a doctor physician that applied ideas of the germ theory to surgery. Remember the work of Semmelweis in Hungary in the 1840s. If a doctor would wash their hands, childbed fever was prevented. He knew that phenol would kill bacteria. Put two and two together, treat wounds with phenol and no infection. Phenol is the basic agent of today's Lysol. So the scientist Joseph Lister started to prevent the microbes to enter our body. This is the phenol experiment he proposed. The first aseptic use in the surgery chemicals used as agents on tissue before surgery also proved that microbes cause surgical infections. So this is the Semmelweis surgery theater. You can see Semmelweis in Hungary antiseptic surgery. Next scientist we can also know the Alexander Fleming. He is the one who started antibiotic. Earlier we can see microbes disease chemicals against microbes. Now we can microbe a product of microbes against the microbes. That is antimicrobials. This work started by Alexander Fleming. We can see you might have studied Alexander Fleming. He discovered the penicillin. Penicillin is antibiotic for fungal disease. He is the one. He is a Scottish physician and bacteriologist. In 1928, observed mold, that is a fungi, growing a bacteria culture. There was a ring of clearing around the mold where the bacteria didn't grow. The mold was later found to be the penicillium species, and the naturally secreted chemicals was called penicillin, an antibiotic. Antibiotics are natural agents, synthetic drugs are chemicals produced in labs. Problems with them, toxic. So these are all the problems. To avoid these problems, antibiotics are natural. He experimented with a petri plate full of microbes, bacteria, and bacteria he spotted. He inserted a drop of a microliter of penicillium collections, penicillium culture. Then around the penicillium colony, there is a clean band around the penicillin colony, which interpreted by Alexander Fleming is that the byproduct of a penicillium is antagonistic against the bacteria. You can see the plate. You can see clear zone of any vision in the plate around penicillium. Beyond that, you can see the full long culture of bacteria. Left side, you can see Alexander Fleming. Then Salk and Sabin in the 1950 they started polio viruses for vaccines for many of the viruses. So how to get vaccines? Avery and McLeod, the two scientists started a vaccine by producing myeloma cells of a rat. Then again 1953 53 to 55, the proposal of DNA by Watson and Crick. You can see the scientists of Watson and Crick. The older man is Crick and the younger man is Watson. This is the double helix. Salk IPV. And they are polio vaccine. He is the one who developed the polio viruses. Isabina. Then later, 1965, Jacob and Monod, they started doing research after DNA, that is RNA and protein synthesis. And the terminology, so you used to coined the terminology transcription, translation, like this, which we can study in biotechnology and molecular biology later semesters. All these things are comes you can see these things in the textbooks. Microbe hunters, Paul D. Cruyff, or Prescott microbiology, or microorganisms, Brock's microorganisms. These are all the reference books. You can see all this history of microbiology. In the next class, we can see the classification and other things, other subheadings of microbiology and plant pathology. Thank you.